motion spectrum and there's an action spectrum. So the action spectrum, I don't know why I have such a hard time saying that, of photosynthesis was first demonstrated in 1883 by Theodore W. Engelmann. Anyway, he took different segments of filamentous alga, which is just algae that's very long and it's stringy and it looks like filaments, and he exposed them to different wavelengths, and the areas that received favorable wavelengths produced excess O2. And so he used the growth of aerobic bacteria clustered along the alga as a measure of O2 production. So he wasn't actually measuring the O2, he was simply using the growth of bacteria as an indicator for how much oxygen was actually produced by this filamentous alga. Again, here we are. Okay, so here, let me show you a big one here. Alright, so at the top we've got the absorption spectra of chloroplast. So you can see that chlorophyll A likes this violet-blue light. Chlorophyll B prefers bluer light. And then there's a second peak you can see along the right-hand side, which we will explain in a minute. And then for the action spectra, which we measure by CO2 release, not CO2, oxygen. The more oxygen is released somewhere around 420 nanometers, and then there's another peak again around 680 nanometers. And if you look at Engelmann's experiment, you can see that he had roughly the same results here by looking at the growth of bacteria. So chlorophyll A is the main photosynthetic pigment, but there are other pigments called accessory pigments like chlorophyll B, which is also used for photosynthesis. Then there are other accessory pigments like carotenoids, and they will absorb light that would otherwise damage the chlorophyll. So they're important because A and B are used in producing our sugars, releasing oxygen, and the carotenoids help to just basically protect everything there and keep it working. Okay, so here's how it happens. When a pigment absorbs light, it goes from a ground state to an excited state, which is unstable. So the excited electrons fall back to the ground state, and then they give off a photon, which is kind of an energy packet, if you will, which is an afterglow that causes an afterglow called fluorescence. If illuminated, an isolated solution of chlorophyll will fluoresce, giving off both light and heat. Pretty cool, huh? Oh yeah. Plants aren't so boring now, are they? Yes, Sarah. They lose their energy. So in this case, the energy is released as a photon. So this shows us here that we have light energy acting on our chlorophyll molecule. Okay? And the electron gets excited. So you can see up at the top the arrow goes up, and as it releases heat and photon, it falls back to the ground state. So when this happens, it causes the solution to fluoresce. So as you can see, B is very fluorescent. Not by today's standards, perhaps, but fluorescent nonetheless. Okay, so this actually occurs in what we call a photosystem, which is a reaction center complex associated with light harvesting complexes. Now, don't get crazy. A complex is just a bunch of proteins that work together. So we've got two complexes here. One that harvests light and one that is the center of the reaction. So a photosystem is going to consist of a reaction center complex, which is surrounded by these light harvesting complexes. And the light harvesting complexes have pigments that are bound to proteins, and they're going to transfer the light energy, in this case the energy of photons, to the reaction center. Okay, there is a primary electron acceptor within the reaction center that's going to accept the excited electrons. So it's accepting those electrons. Leo the lion says grr. So it's reduced as it gains these electrons. So the transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. Did I have you write the entire slide here or something? Well, I obviously was not planning on recording myself when I made